Good afternoon, everyone. Thank you once again for joining us for our continued AMS seminar series. I'm Jonathan Plett. I'm the president of the AMS. And I'd like to start out today in the spirit of reconciliation. The AMS acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respects to their elders, past, present, and extend that respect to all Aboriginal and Torres Strait Islanders people today. Now, it is my great pleasure to welcome our speaker today, Dr. Mary Hanson. She got her PhD from the University of Warwick and was a lecturer then at the University of Worcester in the UK. And then I would like to say she saw sense in not wanting to freeze the rest of her life, and she moved here and is now lecturer and science learning academic coordinator at Edith Cowan University. And throughout her career, her work has totally been up in the air. She's been looking at pollen counts and patterns, detecting pathogen movement, and she's even been looking at trees. And one of the things I found also interesting, she was looking in car cabins at filters and tracking different bioaerosols and allergens that could be blowing in our faces as we sit there in traffic. So today she's going to be talking about one of her recent works, looking at climate change and how it is altering atmospheric microbiomes. With that, Mary, take it away.
Thank you very much, Jonathan. Thank you for that introduction. It sums up what I've been doing really well. I have had a very mixed background. I'm just going to stop my video just so you can see the slides sufficiently. There you go. Hopefully that's still okay. Yes, so today I'm going to talk to you a little bit about atmospheric fungal populations. As Jonathan said, I've had a mixed background, and it's taken me on a little journey to get to this point of looking at fungal communities in the air. I started off in plant pathology, looking at a soil-borne plant pathogen. My PhD was in club root disease of vegetable brassicas. And since then, it's evolved. So I've worked through plant pathology research groups and then ended up getting involved in an aerobiology research group, which is where I became more aware of the microbiome, or mycobiome in this case, in the atmosphere. So just, there we go, sorry. Just begin with then, I just wanted to give you a bit of an introduction to what I'm talking about bioaerosols. You're probably all already familiar with them. So this is just kind of really a recap of where the discipline is at the moment, if you like, because bioaerosol research tends to sit across a wide remit, really. It covers both plant biology, atmospheric science, and also fungal biology and ecology as well. When we're talking about bioaerosols, we're talking about biological material that's in the atmosphere. So this could be anything from pollen, fungal spores, bacteria, plant debris. More recently, people have started looking at viruses as part of the bioaerosol community. Traditionally, they're very hard to quantify, so there hasn't been a huge amount of research done in that on a general aerobiological perspective. So viruses are an up-and-coming area, really. Pollen in the atmosphere is very well established. There's been a lot of research done on pollen, obviously relating to allergens, allergenicity, hay fever, rhinitis, all of those kind of things. So there's been a lot of interest in aerobiology from that point of view. From the fungal point of view, though, the area of aerobiology isn't that well developed. There's been quite a bit of research onto Alternaria and Cladosporium fungal spores, simply because they're allergenic and they're commonly found in the atmosphere. So often when researchers around the world are putting forward pollen counts or predictions for allergen, such as the pollen forecast, they will also include Alternaria and Cladosporium spores because they're the most allergenic. But beyond that, nobody's really documented or created a science out of fungal communities in the atmosphere. And the technology is now present that we can do this. So The exception to that, I suppose, really is plant disease transmission, and that is really where a lot of fungal spore dynamics have been studied, particularly in regard to crop diseases. So to sum that up, really, aerobiology, when we're talking about bioaerosols, we're talking about all of these different biological materials, but some have been studied, such as pollen, in more related to human health, whereas others, such as fungal spores, have been more studied relating to crop diseases. So we've got all of these mixed samples in the air, and then we have to capture them. And you may already be familiar with some of these types of techniques, but more and more they are getting superseded now. Some of the common methods are Burkhardt traps, Burkhardt spore traps, as you can see on the right-hand side of this slide. That's basically sucking in air through the sampling port, and then it can deposit either onto a coated microscope tape, a coated sticky tape, if you like, which can then be cut into daily sections and put onto a microscope slide to see what spores we've got present, or what pollen in a lot of cases. Or it can actually deposit into Eppendorf tubes as well. And that's where we've found that if we're taking what we're capturing into the Eppendorfs, we can then look at DNA. And that's really where my area of expertise lies, and I'll come on to that in due course. You can also capture air samples through cascade samplers. That's shown on the bottom left picture, where there's a series of different pore sizes, and you can put plates in, and it will go through these different pore sizes that are stacked on top of each other and deposit onto plates at different size particles. So it separates by particle size, basically. You can get personal samplers as well, and these are used more in protection when people are looking to see personal exposure to any hazardous, uh, often chemicals or particulates, dust, but also allergens in some regards. So there's lots of different ways we can capture air samples and in order to look at them and see what's going on in the bioaerosol community. And then there's other aspects that we have to consider when we are setting up sampling programs, is to look at kind of what type of transport mechanisms there might be, what type of emission sources there are surrounding our samplers. So what's the land use surrounding the samplers? Is there a particular, say in the case of pollen, I will come on to fungi more shortly, but just to give pollen as a brief example, if you've got a large grassland area, that's going to be emitting a lot of grass pollen during the flowering season. So that would be classed as a local emission source. You also have to think about where you're going to site your samplers. Do you want them at ground level? Do you want them how high above sea level? Do you want them on top of a mast? Do you want them on top of a building? Some people are going as far as using drones and planes to capture air samples now. And this, again, is all to look at various different pollen and fungal spore communities in the atmosphere. So that's what these kind of technologies are being used for the purposes of public health or crop disease or wider plant pathology. Now, the planes is an interesting one. I've read about some studies recently where people are actually creating automated samplers that will project out from the aircraft wing, sample the air for a period of time, then come back in, and then the tubes can be changed over. So you can sample at different heights within the atmosphere. At ground level, you're more going to be getting local emission sources. Conventionally, for long range forecasts, you need to be above 10 meters high, really. So a lot of the Burkhards that are used in long range forecasting and the pollen counts and where they're counting for Alternaria and Cladosporium, they're going to be put on top of buildings. Whereas for crop pathology, you might have them actually at ground level sited within the fields. So I've probably really covered this already, but the main applications of aerobiology are pollen forecasting and crop monitoring. Just an example in this table here. These are some of the types of diseases that can be caused by airborne fungi. So this is why fungi can be a very important but understudied concept in aerobiology, if you like. You've got lots of different rusts, mildews, any kind of leaf spots, scabs. These can all be caused by airborne fungi. Conventionally, these, as I've mentioned, will be studied within the crop and probably individually as well. Most of the time, you'll look at one disease within one crop. And this is actually my old institution, the University of Worcester, in the centre. This is my colleague Beverly Adams Groom, and she was actually responsible for doing the pollen forecast in the UK for many years. It's advanced just recently, so I'll mention that in a moment. The newer technologies have come on board, so they're more automated now. But she's still heavily involved in aerobiology research, and she's particularly interested in fungal allergens, so cladosporium, alternaria, penicillium, all the different types of fungal spores that she might find. But she's an expert in microscopy, so determining the differences between spores and being able to identify which species they are or which genus they are. She's very, very good at that. So I've worked closely with her when we've been looking to compare the results from microscopic counts versus DNA samples, trying to validate the two results. Microscopic counts of fungal spores and bioaerosols more broadly are kind of the conventional way of looking at aerobiology and predicting what's going to happen, looking for trends over seasons, looking to see whether there's any impact of climate change. All of those kind of parameters have been done extensively against data that's been collected onto microscope slides. So microscopic counts going through, very, very time consuming, going through counting slides over a period of, say, a few months sampling period. You're counting daily slides. And that can be resource intensive. And it doesn't obviously give you any data in real time. So there's been quite a few advances in technology recently, especially in regards to laser optics, AI, and also the molecular identification. So the molecular identification is more where I'm working, and that's the area I'm most interested in. But some of the laser optics and the AI, I'm not a specialist in at all. I wouldn't pretend to be, but I've worked with people who are and who are developing those areas, and it's really fascinating. There's a lot of the laser optics they're relying on cytometry and holographic projections, and they generate spectra for different pollen. That's where the focus is being so far. Some people are starting to develop this for fungal spores as well. And it will be fantastic as we get these technologies develop more. You have to calibrate the equipment to each type of particle that you want to look at. So obviously at this stage, the technology has really been focused on pollen. People haven't trained it for fungal spores that extensively yet. People are starting to. So you'd have to expose the equipment to the fungal spores that you wanted to study, and then you'll get a unique spectra for those spores, and then be able to site the sampler out in the field, and it will detect in real time or near real time. And so you can imagine with that, there's a huge amount of data that gets generated. And so that's why a lot of the AI, the machine learning, the classification algorithms, they're all in development at the moment. And as I said, largely for pollen, but fungi is an up and coming area. And so I'm hugely excited about where that's going to go in the future and how we can use that for more accurate crop disease monitoring and so on. Then coming on to the molecular identification, this is probably a little bit more established than some of the new laser optics and AI technologies. But the advances have really relied around metabarcoding and metagenomics. And since this has come on board in recent years, it's started to supersede the traditional microscopy as the way of looking at fungal spores in the air. And it goes beyond what would normally our traditional molecular identification. So where we would use PCR and qPCR to identify different pollen and numerous fungal crop pathogens. So using ITS primers and other metabarcoding primers, we've now started to develop an idea of what the relative abundance of bioaerosols are within atmospheric communities. It's still relatively new, so we're talking really most of the studies are starting to be published within the last 5 years. And it's really focused on metabarcoding. Nobody's really taken it as far as metagenomics yet. So that could be an interesting area to develop as well. The example I want to talk to you today is part of a metabarcoding study that we performed on atmospheric samples as part of a UK government tree health initiative. So the Tree Health Initiative was a large consortium that was established around 2015 through to 2018. Some of the projects went on a bit longer than that as well, but it was to study a range of tree pathogens such as Phytophthora ramorum, which causes sudden oak death, and Hymenopsyphus fraxineus, which is ash dieback. And this was at a point in the UK where we didn't have ash dieback, and we were concerned about the spread. There'd been a few incursions onto kind of the Kent coastline, so the easterly side of the country. And we got involved, the research group I was in got involved in this tree health initiative to study the potential spread of ash dieback across the UK, if it came across, which it was inevitable really that it would. So ash dieback is a fungal disease. It causes dieback of ash trees, as you can see in these pictures. So the crown starts to lose all the leaves and dies back, and the raceses, you can see the leaves start to shrivel, and eventually they will die off. And ash trees are a hugely important component of the UK landscape. So there was quite a bit of concern that the landscape would undergo the same kind of changes that it did when Dutch elm disease took hold in the UK. So we were worried that a lot of ash trees were going to disappear, basically. We had no idea how much genetic resistance there was within the native population of ash, and there's several different varieties. And at this point, we just didn't know what was going on. We knew there was a potential for it to come across from Europe, and we could see what had happened in the European populations. And it was spreading across from Denmark and Northern Europe, across the North Sea, and hitting the east coast of the UK first. And we needed to look at how it was going to spread, or try and predict how it was going to spread across the rest of the UK. So we started sampling for ash dieback, Hymenopsyphus fraxineus, and initially we were looking for qPCR, but we weren't really getting very successful results, probably due to the low spore numbers. There wasn't enough Hymenopsyphus in the atmosphere for us to be able to quantify it. So we set up the sampling campaign over 3 years, and we built it into more kind of atmospheric microbiome study. We set up a rural and an urban site in Worcestershire, and we collected our air samples. These pictures here, we've got Worcester itself is, some of you may be familiar with it, but it's one of the smaller cities in the UK. Really, it's a large town, a small city, basically. And it's still quite rural generally. It does have an urban centre, but it's by no means big, but there's an established urbanisation there. It's surrounded by countryside, a lot of agricultural use, a lot of pasture land, and a lot of arable crops. So we'd got one sampling site in the urban centre at the university campus, actually, and another in the rural location, also at a rural university site. So we sampled each site, the rural and urban location, for 3 years. So this was air samples. So we had them at fairly low height here. They were about 2 to 3 meters high. So we were collecting local emission. We weren't looking for any long range transport with this study because we wanted to look at ash trees that were nearby. We were monitoring them for symptoms of disease, and we were looking to see there were reports that it had been established in our local area, ash dieback that is. So we were monitoring the ash trees, recording cases where there was incidence, and then trying to capture the spores and do qPCR initially. but also we collected DNA for metabarcoding as well. We used ITS2 primarily for that study, but we did do some follow-up studies with ITS1 as well. And these were our sampling setups, basically. So you can see they're not that high. We'd got our sampler on top of a pole system in the rural site and on top of a building at the urban location. So we were capturing our DNA samples into Eppendorf tubes, and we extracted the DNA as normal methods, and we sent it off, and we looked at the ITS2, so that's the internal transcribed spacer region. We looked at the ITS2 in the first instance, and we assigned the taxonomy based on the UNITE database, and we looked at differences between the urban and rural sites. Now, we found that alpha and beta diversity measures did show that there were some differences between the populations. And so we wanted to study these in a bit more difference. Also, there were in a bit more detail, sorry. And also we wanted to look at how this had changed over the 3-year period. Was it impacted by long range variables, such as weather patterns, or was it more dependent on local variables? The key things we found were that actually, yeah, it's local variables that were going to be important. And at the sampling height that we were using, that's not really surprising. You can see in the figure at the bottom here, the air masses in the 3 years were all coming and hitting the samplers from the same direction. There was no huge variation in air mass hitting the samplers, and so therefore any variation we were seeing was likely to be from a local origin. We did find some difference in the top 10 genera that were present, so it altered primarily in the 3rd year, which was 2018. So that was when we saw our biggest change in community structure within the atmosphere, within the atmospheric microbiome at both rural and urban sites. You can see in the plot on the right, Alternaria is the first bar, and you can see in the final year, 2018, the amount of Alternaria dropped considerably compared to the first 2 years, and so on. So there were some differences between years. There were differences between urban and rural sites. And we related this back to weather factors and meteorological factors as well, and we found that leaf wetness was the most associated with the altered populations that we were detecting in the atmosphere. And we also found that human interventions such as harvesting had a key impact on the community structure that we were picking up as well. So overall, we were looking at, because we wanted to focus on the fungi, the economically important fungi, shall we say, so those are the crop pathogens. We looked at Alternaria, Mycospirella, and Cladosporium. They were the most abundant. We found that there were several crop pathogens present overall, several yeast species. What I thought was really interesting was the fact that we were picking up lots of lichenized fungi as well. So we were actually picking up the fungi and the algal partners with the sequencing. We also picked up several non-crop plant pathogens as well. So somewhat expectedly, the crop pathogens that we were picking up were the kind of traditional ones that you would expect to be associated with grasses and cereal crops, because that was the most abundant land use around the rural site, for example. So we'd get a lot of powdery mildews, a lot of rusts, Alternaria, as would be expected. But we had a couple of different species, and I'll mention that momentarily. So looking at the annual differences, we found that there was very little variation between the atmospheric microbiome community in the first 2 years. So 2016 and 2017, they were very similar. The 3rd year was actually drier. And what we found was that the key period it was drier was during the latent period. So that's prior to spore production. So because there was this reduced period of vegetative growth, there's less moisture around. So that, along with that, we observed a reduction in key species and an overall change in species composition. It wasn't that there was less fungal spores in the atmosphere, it was more just the composition of them had altered. So we did record things such as increased Fusinia coronata, which causes crown rust on cereal crops, and periconia, which can cause leaf spots on various hosts. That's not necessarily considered to be that economically significant. Both of those pathogens were generally widespread in the UK. So again, not an unexpected finding, but nice to actually see it demonstrated. We also saw, though, a decreased coniotherium glycines. Now, this is a red leaf blotch of soybean, and actually it's a non-UK disease. It's usually found in sub-Saharan Africa. And we also recorded Paraphoma fumeti, which is conventionally soil-borne. So the this was interesting. It shows really, I think what the key take home from this is that actually, why are we getting these species that we're not necessarily expecting in the UK? And it's cautionary tale, if you like. At this point, this was only with ITS2, so it demonstrates that you need to use more than one marker. And at the time we were doing this study, it was fairly early days. We were exploring, this was our research group's first branch out into metabarcoding. So we'd only included the one marker region in the first place. And the lesson we learned from this really is that you would need to back that up with at least a secondary marker region. So ITS1 and ITS2, or use a different marker such as MACK, TRN, and so on. Some other marker, so you can look at the sequence in some other region of the genome to validate your findings. I still think the results that we've found from this are interesting and informative, and especially for the species that are in greater abundance and where there's more sequence available in the databases as well. But there's one or two results which would need further study to confirm whether they were actual results or not. I mean, it's not beyond the realms of possibility that a soy pathogen could be present, because soy has recently taken off in the UK as a crop. There's lots more soy being grown, but whether it's actually susceptible and whether that means the pathogen is present is debatable at the moment. Nobody's actually studied it in more detail. Between the rural and urban site, we also obviously found some differences. We found tan spot and a leaf blight, Didymela, were more abundant at the rural site. And again, that's going to reflect the agricultural and horticultural provenance in that region. We also found Exiphalia eucalyptorum at the rural site. And again, that was quite one of those findings that I'd question. And so that's why it's got a question mark next to it. However, it is interesting to note that there are actually quite a few eucalypt trees surrounding the rural site. You don't necessarily think that we have eucalyptus in the UK all the time, but there are a surprising amount. And once you start to notice them, you start to see them everywhere. But there was a little clade of eucalypts, and then one or two dotted about. So again, probably could a finding worthy of further study, but I wouldn't say for definite that it was present. You'd have to back it up. Another interesting one that was, we were finding rose pathogens, so rose powdery mildews were more abundant at the urban site. And that would fit with the kind of garden structures that you have in the urban gardens in Worcester. It's quite an old city, and a lot of the gardens are based on that traditional kind of, you might have heard it as an English country garden where you'd have lots of roses present. So the fact that that was present more at the urban site could be a genuine finding. Another one, which was another thing which I thought was particularly interesting, as I mentioned before, was the lichen. We were finding lichen that were resistant to air pollution. We were finding them more abundant at the urban site. And so that then obviously you can use lichen as indicators of air quality, and so looking at the pattern of distribution in the atmosphere as well of the fungi and the algal partner, that could be an interesting area for future studies. We also recorded some. This is the tan spot, I mentioned it earlier. So the one thing I will just note about the tan spot, which we were finding, is it is found frequently across Europe. It's less frequently reported in the UK. It can be found, but when it is present, it doesn't tend to cause disease as much. So whether this is an example of where a pathogen is actually part of the kind of atmospheric community in the UK, but it just hasn't got the right infection conditions to cause disease in the UK. Whereas in the Europe, you've got slightly hotter temperatures, for example. Maybe that could be one that might be worthy of watching in the future for changing climate. Maybe changing climate will alter the infectivity of tan spot in the UK, or maybe it comes down to pathotype. So there's lots that we can study. And I think what I'm trying to convey with this talk really is that there is, just even on this simple study that we did, we found that we were getting so much data that we weren't able to even report it all correctly. We weren't even able to go into the depth that we would have liked. And so there's so much scope for future studies in this area, looking at the atmospheric microbiome communities, that it's hugely, well, I think it's hugely interesting. Just a couple of the other points that it's worth that we found. We found a really clear pattern with harvesting. So you can see in this graph here, the arrow indicates the point at which harvest was completed, because we also did a sampling over a whole year as well, taking weekly samples. Well, not over a whole year, over an 18-week period during the harvest period. We took weekly samples. And there was a clear distinction between harvest communities, atmospheric communities, and pre-harvest atmospheric communities and post-harvest atmospheric communities. This is just the top 10 genus here, and you can see that there's been a change in the abundance. So there was some clear relationship. Obviously, it could just relate to the pathogen life cycles on the plants, but it was correlated so much in time with the actual period of harvest for all of the major cereal crops for various different pathogens, that it seems that there is some mechanism there. And it makes sense, really, does it, if you like. It's just a nice finding because nobody had really documented it before. But for Alternaria, for example, you can see that's the largest peach coloured bar in this graph. When it drops off after harvest, obviously all of the cereal crops where the Alternaria would have been found, they've been harvested and removed from the location, so there's going to be no more release into the atmosphere. So it's probably quite an obvious conclusion, but it was nice to be able to document it. Now, just before I go on to Grassland Circle, the one thing I will just say is that we did actually manage to find some Hemenocyphus, but we couldn't actually identify it down to species level. So because this whole project started off being designed to look at ash dieback disease and how it was spreading across the UK, I think the conclusion was that the number of ash Hemenocyphus spores in the atmosphere were very low, especially in the area that we were sampling, even though there were some indications of disease on surrounding trees. And we could pick it up by metabarcoding, but again, it wasn't confirmed at species level, so it would need a little bit more work to be able to develop that area. But at this point, I should also just give the outcome of the situation for the UK ash trees. It actually turned out that there's a bit more resistance, a bit more native resistance in the ash trees than was anticipated. So that was a great finding. I mean, it's still a concern. It does still cause this damage to ash trees, but on the whole, there are resistance. There is resistance within the native tree populations, and they're capitalizing on that in order to make sure that there is in future planting stocks and so on. So just to then lead on very briefly into the work that we did afterwards. This was again at the rural site, and so we at this point we were interested. It was kind of like, oh great, we've got all of these this atmospheric, we've got all of this information on the atmospheric community. How far are things traveling? Where is it all coming from? What are the emission sources? So we had a circle of grassland set up that was unmanaged. It was left to its own devices largely, and we set up a sampling tower in the middle of it. And this became a PhD project for Godfrey Apangu. So Carsten Scott was a research group leader, and Godfrey Apangu was studying alternaria within this grassland circle. There was another PhD student called Carl Frisk who was looking at grass pollen from the circle as well. And basically, they were both studying emission and dispersal ranges. So Godfrey was looking at alternaria spores, and Carl was looking at grass pollen. And what Godfrey was able to find from his studies in this area was that the grassland was actually emitting fungal spores, and Carl found it the same for the pollen release, for less than 300 meters. It wasn't going that far from the source. So Godfrey demonstrated localized alternaria transport and a strong association to local wheat and canola as well. He did find, because at this point we were now using two marker regions, so we were using ITS1 and ITS2, and he did find that he could get greater taxonomic resolution. Alternaria traditionally is very hard to distinguish down a microscope, and so he was able to capture some of that variability. But again, more marker regions would be great, or maybe even getting into some metagenomics. So future work, how does all of that relate to what I'm doing now in at ECU? So I came over from the UK earlier in this year, and that was the latest work that I've been doing there. And I've come over, and I'm hugely excited to start this kind of work in Australia as well. So hopefully we'll be able to get some studies up and going of the atmospheric microbiome in WA and other states. It would be great if we could get some comparisons between different areas. It would be lovely if we could get some laser optic samplers to monitor important fungal pathogens or allergens in real time. This could be particularly useful for key diseases. And then if we can combine that with, say, metabarcoding to get a really good insight and a network of pathogen monitoring over local scale and regional distributions as well, that would be fantastic. The only problem is the laser samplers are still fairly expensive. Hopefully they will reduce down in cost relatively quickly, so they're more accessible. But I think one of the key things from the work that we've found so far is that we need more molecular markers, and we need to achieve that greater taxonomic resolution. And I'm from the sequencing data, and I know that is a problem that a lot of people are facing. So hopefully there'll be huge advances in that in the future, and we can look at like low coverage genome skimming and that kind of thing to get more of an idea of what is going on, and be more confident when we're in determining what members we've got, what fungal spores we've got present in the atmosphere. So I'd just like to acknowledge the research group at Worcester, and I'd also like to acknowledge my new research group at ECU, in particular Anna Hopkins, as her involvement has been great in helping to get me established and familiar with everything that's going on in Australia, and to all of you listening as well for allowing me this opportunity to present. And there's a few references. So thank you very much for listening. And has anybody got any questions?
Great. Thank you very much for that, Mary. That was awesome. So there are already a couple questions in the Q&A. The first from Nicola Day. Do you know if the fungi are always present in spore form in the air, or if they're hyphal fragments as well?
Yeah, that's a really good question. What Godfrey found, Godfrey actually was finding hyphal fragments as well. So it wasn't just the spores, and that's an area that, yeah, that definitely needs further investigation to see what's going on. We found it with again, sorry to keep going back to the plants, but we were finding leaf fragments in the atmosphere we were detecting plant DNA at an abnormal time of year as well, and so we questioned that. It wasn't when you'd expect pollen to be out, and we found that we were actually getting leaf fragments. So yeah, basically any small fragment of biological material seems to be there.
So how would you actually um, try and balance that, right? Because if you're having spores, they might have less DNA than
Yeah.
whole fragments. Do you have any ideas on that yet, or that it's just so new that you don't?
Not yet. I don't. It would be great to be able to quantify it. That's why some people are kind of dismissing the microscopy a little bit as being a bit old hat, but that's why I think the microscopy is still going to be really useful. And maybe that's where the filters and larger sampling, 'cause you mentioned my car cabin filter work earlier, but maybe that's where, if you're actually capturing onto larger filters and then extracting from the filters, if you can see exactly what's going on and get hyphal fragments from them as well, that might be a way to go about it.
Cool. Bevan, you have a question.
Thanks. Yeah, long that I couldn't be bothered to type it in. Awesome talk, actually. It was really
Thank
cool.
you.
I think relating to the other questions is fungal spores are like incredibly variable. So you've got spores, and you saw this in your work. So rusts are often specifically adapted to have that kind of flat shape, and that they're almost they're adapted to be aerobically dispersed. Then you've got sort of dry spored ones like Aspergillus cladospore and that type of thing. And then you've got other types of spores that I've done a bit of work on, say Mycological, which are hyaline and quite soft, and they're typically considered to be like a splash dispersed. So you've got these wide range of different spore types, and like, how do you account for that, or you just kind of like accept it and you're monitoring change rather than sort of absolutes?
At the moment, we were just monitoring change. That's a very good point. It's something that would have to be accounted for in an in-depth study. So yeah, any ideas about doing that in future work would be gratefully received.
Great.
That's certainly a good point. That there's loads of different factors, because there's so many unique aspects to different spore morphologies and dispersal rates and so on, that I think this is really an area that I think it warrants a lot more attention. So hopefully, whether funders agree, that's another matter. But
A question from Tracy. I'm sorry if I missed this in your talk, but did you test for or notice a seasonal effect on the number of detections for H. frax or any other pathogens?
we didn't manage to get enough of it. We couldn't pick it up enough. We'd probably need to have it. There was another site on the south coast of the UK which had a lot more ash dieback, but at the time it ended up with being to be more heavily diseased. So we tried to get samplers down there, but we didn't get it arranged in time for the project. So we didn't detect enough to be able to draw any conclusions on it. But I think other researchers might have taken that on further since.
Great. And Tracy would love to meet up with you at APPS if you're going to be there.
Okay, awesome.
Because she's also going to be presenting work done with airborne fungal work. Tom May says, great to know that you'll be continuing your research on fungi in Australian air. What do you know about the viability of the spores?
Generally, or so, because, well, again, it could vary depending on species. It could, again, it's something that could be tested. Most spores are going to be pretty hardy. Sorry, light's just gone off in my room. Most spores are going to be pretty hardy, but we haven't tested it. I think you'd have to probably go through, and this is where I think a lot of the work is missing in a way at the moment. It needs putting together. So people will have studied that for various different pathogens. And so then we need to go in to see what exactly is present in the community. I'd say a study like what we did, set up over a few years, looking at different marker genes. So we're very confident in what we've got present, looking at what's going on in the surrounding environment, and actually proving Cox postulates for all of the different pathogens that we're getting present as well, because there's some that we're not going to even know about, and some surprises that we're not expecting to be there, but maybe they are present all along. And then doing things like looking at how long spores are surviving as well, and getting that seasonality over a whole year. It would be great to do it over several years. Say 5 years of consistent sampling would give you a fantastic data set, especially if you were looking at ground truth as well.
So following up on that, you've mentioned a number of times, like the red leaf blotch and others, didn't expect. So how are you thinking of bringing in a biosecurity aspect to the research that you're doing, or is it, can you comment on that?
It could be useful. I think once you know whether it's an accurate result, I think that would be the thing first. You need to have confidence in the methods first. So now the metabar coding methods it's well established now that you need to use more than one marker gene. You need at least 2, don't you? So once you've got studies going on that are doing that, then it could certainly have a biosecurity impact. You could use it as a monitoring or a surveillance kind of system. The technology is still a bit pricey for that, for the laser optics, because that's how you'd have to convert it to real time. You'd have to validate the laser optics with your microscopy and your DNA. But then once you've got the laser optics up and running, then that could easily be a surveillance system, a biosecurity surveillance system, but you'll need to figure out how frequently you need samplers located. At the moment I think about $100,000 each, if not more, for some of the good ones. And
when you say a bit pricey, that's very pricey.
yeah, so the cost needs to come down. You need a lot more people putting a lot more different spores through them to get the algorithms and train them, and then eventually, there could be a nice real-time surveillance system in place that would be monitoring air samples. That would be ideal, especially for somewhere like WA and Australia as a whole, because in places like the UK, it's kind of a bit harder to do that biosecurity actions. Because there's so much trade and we're so close to Europe, it comes across really quickly. Whereas because Australia has that capacity for that biosecurity, it would be an ideal place to test something like that out long term. That would be exciting.
For our last question today, then, Tracy also has another question for you. Go ahead, Tracy.
Hey, so it's interesting you're talking about the biosecurity side of things. The project I've done that I'll be talking about at APPS is looking at the difference between biosecurity using airborne eDNA monitoring for plant pathogens and biodiversity. And
lovely.
we've already shown we can detect HVACs on a seasonal basis with higher detections in the middle of summer when you expect it, over multiple years using a data set over 10 years.
Oh, brilliant.
Monitoring stations across the country of Switzerland. So really keen to chat about this because I've actually did this project specifically for WA and detection of myrtle rust.
Ah,
so
brilliant.
let's chat.
Oh, yeah, that would be great. Yes. And Anna might also be interested in that as well if you talk about myrtle rust, because we've currently got a PhD student. I don't know if you're aware of him looking at it as well. So yeah, that sounds fascinating. I'm not sure I am going to be there, but if not, we should definitely
either way we'll figure it out.
Yeah, that would be that'd be fascinating. It sounds like you've developed a bit more than we had at this stage when we were doing this project, so that's great.
Yep, sounds great. Thanks, Mary.
Perfect. Thanks, Tracy. And that is what we're here for, right? It's trying to connect people as well. So thank you very much, Mary, for today, for the talk, for the information.
Thank you for having me.
Welcome to Australia. It's great to have you here. For everybody else, thank you for joining us today.